Hello and welcome to this free webinar on metagenomics. Metagenomics is a way to study microorganisms in their natural environment, dissecting community structures, exploring species richness, and investigating the functional roles that these microbial communities play in many areas related to both human health and disease. Now, this webinar is provided as an introduction to the topic of metagenomics, and we will also discuss some of the opportunities to continue and learn about metagenomic data analysis. The session will be recorded and made available on YouTube, so anyone watching this will be able to follow up. I'll also discuss several free resources that you can have access to after this webinar. In this webinar, you will learn about the upcoming OmicsLogic Metagenomics program that will be starting on August 31st. This online program is designed to be equally rewarding to both students and advanced researchers, and is especially suited for project-based learning on a flexible schedule. The program will cover main aspects of metagenomic sequencing, analysis of 16S ribosomal RNA, and other aspects of microbiology research using bioinformatics. And at the end of the program, participants will be conducting independent or group projects, analyzing data from the public domain, and learning from high-impact biomedical publications on various topics related to the microbiome. Now, for those that are not familiar with the OmicsLogic data, with the OmicsLogic programs, I want to briefly explain what they are. All of our OmicsLogic training programs include interactive online sessions like today, self-guided study materials that we have a collection of, organized by relevant topics. And these have been carefully designed with practical assignments in mind, so that you don't only learn from lectures and presentations, but you also get to apply what you have learned to example data sets, and then eventually to a project of your own. And as in all of our other OmicsLogic programs, this OmicsLogic Metagenomic program will include all three of these elements. The objective of these programs is to allow students as early as high school and researchers as advanced as faculty to learn bioinformatics, or the data analysis aspect, related to a variety of topics. And so this program is going to be focused on metagenomics. The program is going to start on August 31st. And so in this webinar, you'll get to see some of the examples that we will use in the program itself in much greater detail. And I'll also introduce you to some of the free resources that you can have access to as early as today to learn about this exciting field. So since the discovery of the microscope, humans have been learning about microorganisms that surround us. As the resolution improved, more and more representatives of the unseen world have been discovered. The scale of optical resolution can reveal details of up to 1 micrometer, which is a typical size of a bacteria. But today, microscopes are tremendously powerful, allowing humans to see smaller and smaller elements that reveal how the world of microorganisms function on every imaginable surface and is actively interacting with each other. So in this image, for example, we can see a super high resolution image achieved with helium ion technology that shows the process of bacteriophages, which are colored in green, attack E. coli bacteria, which are blue. So you can see here a resolution expanded and how these bacteriophages are attached to these bacteria. The connection between disease and microorganisms has been established in the 1800s, and already towards the end of the 19th century, laboratory methods for growing microorganisms in a lab culture were developed, helping characterize them with staining. These studies provided important information about bacteria function in isolation. However, it was soon very apparent that most microorganisms live not in isolation, but in complex communities. And today, we know that the microorganisms or microbiota, which are communities of microorganisms that make up the human microbiome, are not just unicellular organisms living alongside each other, but instead form highly regulated, structurally and functionally organized communities attached to surfaces as biofilms with interspecies collaborations, as well as antagonisms that contribute to ecological stability. The stability of these communities is fascinating, as many of us probably know from trying to battle such common skin conditions as eczema, acne, and dry skin. Forming distinct communities, these microorganisms are interconnected and determined to maintain their community intact. Bacteria within a biofilm can communicate with each other by producing, detecting, and responding to small diffusible signal molecules in a process called quorum sensing, which confirms benefit for host colonization, biofilm formation, defense against competitors, and adaptation to changes in the environment. Quorum sensing activities in biofilms are also involved in the virulence and pathogenic potential of bacteria and are therefore an important factor in understanding and controlling bacterial infections, as they enable microorganisms in biofilms to become more tolerant of host defenses and antimicrobial agents. So inside each community, there's a balance of bacteria, fungi, and viruses that work together to control species abundance and activity. The human microbiota is estimated to be 10 to the 13th to 10 to the 14th of microbial cells, with around 1-to-1 microbial cells to human cells ratio. Previously, it was thought that there is a much greater abundance of microorganisms compared to human cells, but since 2016, this theory has been adjusted. These numbers are actually derived from the total bacterial cells in the colon, which is about 3.8 to 10 to the 13th of bacteria. This organ harbors the densest number of microbes. However, each one of these microorganisms carries important functional properties in genes that contain genomic information. These properties and the genomic information about microorganisms have been of a growing interest to the scientific community, resulting in over 80,000 publications that have been continuously being published in the past decade. The majority of these studies have focused on the intestinal microbiome, as you can see right here. But more and more studies are starting to look at other functional communities throughout the human microbiome. Since the 1990s, microbiome studies were significantly advanced through such major initiatives as the Metagenomics of the human intestinal tract and the subsequent Human Microbiome Project. These were huge studies that are ongoing and that have been made possible through the advances in metagenomic sequencing. As a result, we now know that there are distinct communities that can characterize skin, lung, gut, and even brain microbiota. Today, we know that the endogenous human microbial communities contribute to critical metabolic, physiological, immunological, and even psychological functions, including differentiation and maturing of the host mucosa and its immune system, food digestion and nutrition, energy generation, metabolic regulation and control of fat storage, processing and detoxification of environmental chemicals, maintenance of skin and mucosa barrier function, development and regulation of the immune system and fine-tuning of its reaction pattern, which is the balance between pro-inflammatory and anti-inflammatory processes, prevention of invasion and growth of disease-promoting microorganisms, and many more. Importantly, these studies and questions being answered about the microbiome have been as a result of important omics technologies. These include sequencing technologies, sequencing of the DNA and the RNA, as well as mass spectroscopy, metaproteomics, and metabolomics. Together, these studies form a multi-omic approach, which is something that allows us to study the omic data about a community and find associations between different features in distinct omics data types. So in this program, we will focus on one of these omics data types, which is metagenomics. So unlike proteomics, genomics, and transcriptomics, which all focus on the whole of a single organism, organisms in the microbiome live in communities and interact with each other. And so when we study them, we want to study the community and the most dominant and functional members of that community. So the metagenomics approach is an approach to study genetic material sampled directly from an environment, which allows the study of communities of organisms directly in their natural environment and bypasses the need for isolation and lab cultivation. However, metagenomic sequencing of microbiome communities and detailed studies of individual members of that community are typically working together hand in hand. For example, metagenomic sequencing can be applied to environmental culture-free sequencing to study populations where we are looking at culture-free sequencing reads using databases to annotate operational taxonomic units and study taxonomic classification. In the case of culture-based sequencing reads, we are typically using reference genomes and assembly to study genomic classification by understanding how the specific members of the community that we're interested in are different from other known members and how might they be related to each other. In the case of 16S metagenomic sequencing, we can use specific genes, for example, the ribosomal 16S RNA gene, to understand the relationship of different species at different taxonomic levels. What is important about this approach is that the 16S gene is short enough to fit in its full sequence in a typical paired end read for metagenomic sequencing. The 16S ribosomal RNA gene itself is about 1,500 base pairs long, and when it is sequenced, typically only one portion of that gene, a hypervariable portion of that gene, is sequenced to understand how it can represent the complexity and the relationship of the community based on the presence and richness of particular species. This is done by relying on what's called operational taxonomic units, which are basically a definition of a particular type of group of sequences that relate to closely related individuals. Those could be organized by hierarchical clustering algorithms and annotated using specific databases. Some of these databases include the Silva database, which contains taxonomic information for the domains of bacteria, archaea, and eukarya, which is based primarily on phylogenies for small subunit ribosomal RNAs. The taxonomic rank information for archaea and bacteria is obtained from Berge's taxonomic outlines. But there are other databases as well. These include the Ribosomal Database Project, or RDP, as well as the Green Genes taxonomy. It has been studied that the Silva database provides some of the most up-to-date and validated information. As a result of this annotation, we can organize different communities in terms of their biological classification, revealing the composition of distinct communities that could be specific to different organs or compared between different conditions. But the 16S ribosomal RNA allows us to study only the bacteria. However, when we talk about the microbiome, that also includes the virome. The virome is one part of the microbiome which constitutes all of the organisms that inhabit us, including bacterial and archaeal organisms, as well as the microbiome and other members of the meiofauna, such as protists and metazoans. Studies of the virome are in their infancy because it has only recently become possible to see the virome in large sequence data using bioinformatic tools that can detect relationships between viruses despite extreme nucleotide sequence diversity. The size of the mammalian virome is actually not known. Although our own cells are outnumbered about tenfold by our bacterial microbiome, the number of viruses may be tenfold higher still. Human feces alone contain about 108 viruses per gram. Additionally, the mammalian virome is continuously updated through rapid evolution of viruses and through exposure to viruses in the environment or in other mammals. So the microbiome actually helps contain the diversity of the virome, forming a protective layer between our immune system and the environment that we're in. To study the virome, additional types of sequencing methods could be used. Specific libraries can help amplify the amount of genomic information that we can extract from these microbiota and amplify them enough so that we can map them to reference genomes, assemble them into novel genomes, and discover how they evolve based on specific conditions or samples involving diseases. So we know viruses causing infectious diseases that are typically acute. These include flu, dengue, Ebola, and COVID-19. However, many of these viruses also cause chronic diseases that sustain continuous inflammation and lead to diseases even as complicated as cancer. Some of the commonly known viruses that are associated with thousands or hundreds of thousands of cases of cancers every year include the Epstein-Barr virus, human papillomavirus, and hepatitis B and C. So in this program, what we'll do is we will use practical examples from the world of the microbiome, virome, as well as individual species that play major roles in human health and disease. One of the projects that we'll look at is called the American Gut. This is a project that is a portion of the Human Microbiome Project, and it relies on citizen science, which means that every single one of us can contribute a sample from our saliva provided to them, and they will do the sequencing and find how our sample compares to other known samples from a variety of people that also answer self-survey questions. For example, your diet, your genetic conditions, your ethnic background, age, and others. We will also look at specific types of examples that help us understand the function of microbiome in conditions that are non-intuitive. For example, the gut-brain axis might show that there is a relationship between the microbiome and behavior. So in this example, we'll take a look at how researchers were trying to look at the specific function of the microbiome, the gut microbiome, irrespective of the diet itself, that had an impact on anxiety. To study many of these types of data sets, we will rely on bioinformatics. So when we speak about bioinformatics, that includes taking sequencing data and analyzing this data to understand the relationship between sequence variation and particular phenotypes that we can study collecting other types of data. To do that, we will rely on an intuitive platform called the TBioinfo platform. On this platform, we have specific pipelines that have been designed to make such studies intuitive and quick. So let me show you an example. Here, I will just show you a brief example of how you can understand a complicated bioinformatics pipeline that actually produces organized information and visualization about a particular data set. So I will go here quickly to the metagenomics microbiome area, and you can see that I have a couple of sections related to this. One of them is related to whole metagenomic sequencing. And in ribosomal RNA. Sorry, please, when you're joining, make sure that your microphone is muted. So what we'll do is we'll first understand the logic of analysis. In this case, we're dealing with 16S ribosomal RNA, which is sequencing of hypervariable regions in the 16S ribosomal RNA gene. After that, we will apply the data to bioinformatics workflow and understand each step that helps us eliminate the noise and create clusters of sequences called operational taxonomic units. These operational taxonomic units could be then annotated based on their relationship and based on known operational taxonomic units that will reveal the complexity and the relationships between different samples in our study. To visualize, we will use a popular package called phyloseq that allows us to build phylogenetic trees and understand species diversity and richness in comparing multiple types of samples. So let's take a look at some of the outputs that we will get. One of the outputs is going to be a table of abundance for individual OTUs. Since they were prepared based on all of the samples in our data, we can now select specific samples in the data associated with a condition and compare them between each other. So once they are annotated, you will see that specific level of information at the species, genus, family, and order and class could help reveal how different types of diets or microbiomes that were results of these diets could show differences between these two conditions. By providing such examples, we will understand the inputs, the logic, and the outputs of complex bioinformatics pipelines. And as a result of this program, once that logic is clear, you will think about how can I apply the same types of approaches to study some other examples that might be relevant to whatever you're interested in. So what kinds of questions or what kinds of projects could be studied using such analysis? In many cases, we can compare conditions, for example, a healthy versus disease. But you can do even more. By understanding how these studies characterize the function of specific bacteria, you can study a variety of different data sets related to the impact of antibiotics, the relationship between microbiome and different treatments, understanding how to select specific interventions, and many discoveries that were related to microbiome changes associated with conditions such as Alzheimer's disease and others. Especially in treatment selection, we now know that the microbiome has a specific impact on our ability to process different types of treatments for cancer, and even find specific variations in the microbiome that help predict drug response. So by providing such examples, you will be able to apply different skills related to bioinformatic data analysis and integration of different types of omics data to understand how to find microbial signatures, how to understand functional species richness studies, and understand how to find and extract data sets from these public repositories for your future studies. So what we'll do in the program itself, in the OmicsLogic Metagenomic program, is we will spend 2 weeks learning about different types of sequencing techniques, analysis steps, like mapping on databases, data visualization and processing, and understanding how to apply pipelines, bioinformatic pipelines that produce different types of outputs. In the second week, we will start seeing different examples, project examples like the American gut and the human microbiome, the diet and anxiety relationship as an example with the different types of disease studies, and other research examples that are based on treatment variation and intervention. An important part of this program is to reveal how bioinformatics can help us explore big data from different types of omics and find associations between omic features and clinical phenotypes. And when we do this in the context of the microbiome, we will look at the relationship between cellular omics, or the host, as well as the microbiome and the virome, to understand how different types of clinical conditions develop and could be intervened. As a result, you will have time after these 2 weeks to develop your own bioinformatic project. And that will be presented for expert feedback and community feedback so that you will understand how you can demonstrate the skills that you have acquired and translate that into an impactful project that demonstrates both your research capabilities, ability to apply bioinformatic analysis, and develop specific visualizations and presentations that help you communicate both your skill and the research discoveries that you have made. Now, an important objective for us in the OmicsLogic programs is to provide a clear perspective on how bioinformatics, machine learning, and artificial intelligence is used in research, clinical, and industry applications. And what we'll do is we'll do this by quickly providing you with access to industry standard tools and enabling infrastructure, exposing you to a variety of different analysis approaches that clarify the logic in application to real data sets, and provide you with some examples with a variety of projects where you can see how variation in the settings of specific parameters can play a major role in the output from these different studies. As a result, you will understand how different types of sequencing technologies and different experimental designs could be translated into a step-by-step bioinformatic analysis pipeline, and how different types of outputs could be interpreted from a biological or clinical standpoint. So, and our team, our mentor team in the program, will be able to assess your progress at every stage of this developing program, and provide you with some feedback on where you might apply specific types of logic and improve your understanding so that you can become more effective using bioinformatics, in this case, in application to metagenomic data. Now, there's a question here from Christina, who is asking, will there be another offering for this program later this year? So we have a whole yearly schedule of different types of programs that we do, and we'll be able to share that information with all of those that are interested. An important part of this is that we are going to rely on public domain data sets. So we'll actually do a whole session on how to find appropriate types of data, how to understand which data sets are going to be applicable for different types of analyses. And we'll do a hands-on downloading and exploring different types of data sets and understanding what they might be useful for in the context of microbiome and metagenomic studies. Many of our previous participants have been able to demonstrate what they have learned in the context of research projects and presentations at conferences where they were able to provide an insight into a particular area related to their research or to their study, including high school students that have used this as an opportunity to develop research projects in the context of their application for colleges and further studies that they were interested in. So first of all, We will start by doing a little bit of an introduction and an overview, and the program will start on August 31st. And this is just a few sessions that we plan to have. And before I go through these, let me show you where you can find all of this information as I am speaking about this. So if you're interested in learning about metagenomics and microbiome, I'm just going to explain a little bit about how we will structure this program and what are the different resources that you will have access to in this program. So on August 31st, we will start the sessions. The sessions will typically happen between 10 and 11:30 a.m. CST, Central Standard Time. And what we'll do is we'll start with a review of the program objectives, the different topics that we will cover, and the different resources that you will get access to in the context of this program. We will then talk about metagenomics. And here I want to highlight that there is a free course that you can already take on an introduction to metagenomics and introduction to bioinformatics for those of you that are interested but just getting started. You will find those on the same website, edu.t-bio.info. So in the introduction to next generation sequencing for metagenomics, we will talk about how next generation sequencing technology could be used to study whole genomes and study microbiome. We will then talk about how the microbiome is a functional bacterial community. And we'll do this using an example from the human microbiome study. So here we'll discuss different types of databases like SILVA, Green Genes, and NCBI, and talk about what are these hypervariable regions and how are they used to produce operational taxonomic units. We'll then talk about the variability and composition. Ultimately, we would like to compare different communities to each other. So how can you do that knowing that this is going to be composed of different types of units that represent hypervariable regions? So we'll talk about methods like Shannon diversity and species richness, and especially focus on different pipelines that are designed to eliminate a lot of the noise that comes as a result of these specific sequencing technologies. Then we'll transition to our hands-on project sessions. In these sessions, we'll use projects like the American Gut and other examples, and also discuss how you can find some of these freely accessible public domain data sets and apply the same analysis tools that we've discussed in week 1 to the different data sets to ask specific questions from data. Then we'll look at this relationship between microbiome variation that can be established in the early years of life with long-term effects on subtle conditions. For example, these could be chronic diseases, or they could be subtle conditions like anxiety. We'll also talk about how to develop a metagenomic project. So if you're interested in another area, for example, you might be interested in questions like the association between microbiome variation and diet, or the impact of microbiome variation on disease or tolerance to different treatments. We'll talk about how to find data, how to upload the data to the cloud-based systems that we will provide you in the context of this program, how to then prepare your project to focus on a particular question, and follow specific techniques that allow you to record every step of your analysis to make the data analysis reproducible and presentable in the context of a research study. What's important is that at the end, you will be able to submit your project for evaluation. And when you submit that project for evaluation, we will focus on the best projects from the whole program that will get feedback from experts in our network. These experts come from different academic projects or programs, and from industry experience. So they will be able to provide you with both feedback on the project itself, but also answer any kinds of questions that you might have about the future using bioinformatics in application to metagenomic data sequencing. So let's take a look at some of these resources that we've been mentioning. As I said, there is a platform that you will get access to for doing your metagenomic analysis. And this platform provides different levels of access. You can start from demo, which is what I just did now. And I showed you an example of a specific project and the analysis inputs and outputs, as well as the logic. After you complete several exercises in the demo version of the platform, you'll be able to upload specific data sets that we will provide you with that are annotated and already understood by our team so that you can make sure that your analysis is reproducible. After that, if you continue and develop a project of your own, you'll be able to go to NCBI and start researching different accessible microbiome data sets, understanding what types of data sets are out there, how to find the necessary types of data for the analysis that you're planning, and understand different types of sequencing techniques that are going to be possible to use in the context of your research program, of your research project, sorry. In the same time, you will be given specific assignments. If you look at the syllabus, you'll see that for each one of our sessions, we will have specific coursework that you have to complete. Inside this coursework, you will be able to do practical assignments based on a particular project. So by completing those assignments, you'll actually run some analysis and see the result that you will have to interpret from a biological or a clinical standpoint. Now, throughout the whole process, we will have a lot of support available to you. When you are facing some technical challenge or you have a question that you need clarification with about methods or data sets, we will be interacting with you using a forum. And at the end of this process, you will have to submit a project where you identify a specific problem statement and develop an analysis plan for a particular data set from the public domain. So you can see here some examples, and we'll be providing you with examples of previous participants of these programs where you can learn about how they have been able to develop very interesting projects on a variety of topics, from different types of disease studies to an understanding at the basic mechanistic level how some of these pathogens and bacteria function. So this is where you would go to complete your registration. Bipsa already in the chat pasted some links for pre-registration. So the way that the process works is you first submit the information about your current educational background, your interest, your level of familiarity with bioinformatics. We review that information and we provide with different types of suggestions for you to continue on to complete your registration. Once you complete your registration, you start getting access to the resources that will be included in the program. I have to note that these resources might take some time for you to complete. So if you have time before the program starts on August 31st, you can already get started with some of the introductory materials and be caught up for the program itself. You will also be able to get access to a whole community of participants. We have several thousand participants on the platform that all work through similar coursework and similar types of analyses. And then we will facilitate interaction between the participants so that you can potentially organize a group project together with others and learn from others that have more experience. So I will welcome all of you to join us for this OmicsLogic Metagenomic program. Again, the microbiome itself is a fascinating topic that provides many opportunities for research and study. You will be able to understand both the aspects of biological and clinical importance of the microbiome and the different members of the metagenomic community or the microbiota. You will also get access to bioinformatics tools and training that will give you some skills to develop an independent project and apply your bioinformatics understanding to different types of public domain data sets. Now, at the end of this presentation, I just also want to mention that you can already connect with us and our team on social media. You can do this by joining our group on LinkedIn, where there's a community of over 500 students, researchers, and industry professionals that are all engaged in bioinformatics, machine learning, and different types of data science. And you can learn by interacting there with the members of this professional community. We also have a large group of participants of previous programs on our Facebook group, and that is called Learning Biomedical Informatics. So joining that also will give you access to some of the recurring program information where you can keep up to date on upcoming programs. And these programs include much more than metagenomics. We have a yearly schedule of programs related to genomics, transcriptomics, metagenomics, epigenomics, data science, oncology, precision medicine, and infectious diseases. So there's really a variety of topics that you can explore. If you go to our website, you will find that we have several upcoming programs that are highlighted, and also a lot of different resources, such as courses, projects, specialization tracks, and even coding skills that you can gain by practicing different types of data science techniques and machine learning. So we're excited to welcome you. I will open this for questions also. If any one of you has any questions, I would be happy to answer those now.
Hi, this is Shreya. It was a great presentation earlier. Could you tell me where exactly could I find out the information about the different data science courses or the information where I could get some information about data sciences?
Yes, I'd be happy to, and thank you. We have a code.omicslogic.com, which is a place where once you have registered on the EDU site with the same login information, you can practice all kinds of data science related exercises in both R and Python. And learn about how to apply these different types of methods to different omics type data. So code OmicsLogic.com, but you do have to be registered on this educational portal first for your account to be confirmed.
Yeah, thank you.
Thank you. So again, for all of you that are joining for the first time, registration here is free. All you have to do is right here, you will see a place where you have to provide your email and password. You can also register using different types of social media platforms. And what's important here is that you actually get to build your profile starting from free basic courses. So you don't have to pay anything to actually start your bioinformatics training. Here you will go to login or register, and you have a variety of options here. So you provide your first, last name, and register here. I will just log back in and show you a couple of things about your profile. So once you register and have an account on this educational site, you can choose between different types of access. So there are many courses that are free, including Introduction to Bioinformatics, Introduction to Metagenomics, and others. You'll be able to find them here under the courses. And you will start building your profile. This profile includes a point system that allows you to generate points for completing different types of courses, doing independent projects, and participating in different programs on different topics. All of these will then be displayed on your main profile. So let me give you a different example. That's not me. So here's an example of a participant from a previous program that was able to complete a full program and also develop an independent project. This kind of a program actually offers you a full profile with a certificate from the latest program that you have completed, and you can share that obviously on your CV or LinkedIn. As you complete different courses, there is an interaction. So you do submit quizzes and different types of assignments, and you get graded on those assignments. You actually get feedback. If you face some challenges, you'll be able to see that. Our previous participants have provided important feedback about how this program is different from your typical online course. So I invite you to explore our blog and also look at this blog on one specific article that I think summarizes a lot of the different ways that this program is different from your other typical programs on bioinformatics. So importantly, this is not a program where you just look at lectures and go through some videos. There is an interactive session where all of your questions are answered. And there is going to be a feedback that you get for your personal progress in the program. Okay, so there's a question. The best courses for beginners, I would recommend the free courses like Introduction to Bioinformatics and other introductory courses. However, in the program is designed for high school students all the way to faculty. The interesting thing is that you get to participate in sessions and make decisions on your next steps. So you can, again, use the asynchronous courses that cover a variety of different topics, join the program sessions to get answers to your questions and focus on a particular domain, and also then decide whether you are interested in developing an independent research project or not. And so to answer Christina's question here as well, you can either purchase courses individually or select a membership option that will allow us to take course offerings in parallel. Exactly. So as a part of your program, you will have access to particular types of courses. If you are interested in taking other courses not related to metagenomics, you can also get access to those through our basic membership, which starts at $10 a month for premium courses. And for the courses that are free, obviously you only need to be registered. Now, throughout the year, we have different types of training programs that are focused on different types of omics data. These include genomics, transcriptomics, epigenomics, metagenomics, data science, oncology, precision medicine, infectious diseases, and others. And so you also can get an unlimited membership that gives you access to both projects and all of the sessions throughout the year. Importantly, many of the research users that we have are leveraging our cloud-based infrastructure to conduct their own independent bioinformatics research. And in those, they utilize a research account. That research account also gives you access to the cloud-based tools, as well as the sessions that we have for all of our programs, and access to asynchronous courses if you're interested in learning about something specific. We also have a series of projects. These are examples that provide you with a springboard into a particular type of application of data science methods and different types of sequencing data that you can explore here on agriculture, different medical topics, Alzheimer's or different neurodegenerative diseases, and others. Okay, so again, the program is going to start on August 31st. You still have until tomorrow to take advantage of the 30% discount to complete your registration for the Metagenomic, OmicsLogic Metagenomics program. Once you complete that registration, or even before you complete that registration, you can start accessing courses and projects that are available here. How will the feedback help sessions work for this metagenomics course? So if you're interested in developing your own research project, you go through the basic training and start thinking about what type of a data set and what question you would like to answer with that data set. So if you want to take let's say, the impact of microbiome transplant from fecal transplants, the impact on, let's say, obesity, you can find a data set like that on NCBI and then learn in the courses and the interactive sessions how to actually take the data set, apply a specific analysis, and interpret the results. At some point, you will submit your proposal. So at that point, you'll get to present your proposal to both the participants of the program and the trainers in the program and get feedback. They will provide you with written feedback, and you will be able to start working on your project. At the end, when you have completed your project, you will submit a presentation, either PowerPoint presentation or a video. And if it is one of the top 10 projects, you will actually get feedback from an expert. That could be an industry expert or an academic expert in bioinformatics that will provide you with some feedback on what you have done. So Shreya, hopefully that answers your question. Another question, could you please suggest the best course for plant biologists? So depending on your question, I think we have a number of projects like the crop drought stress. But to be completely honest and transparent, I think that the majority of the courses are a little bit more on the biomedical side. If you're interested in plant-specific projects that are related to the microbiome, we will discuss the particular types of sequencing and a few different examples, but you're free to start working on a microbiome to plant related questions. We do have examples of that we could leverage in the program as well. And that's actually what makes this program, I think, more flexible. You could actually look at a variety of different questions and data sets starting from the very beginning of the program. Okay, the question, is there any tool that can be used for interpreting the results? So typically that will include some analytical tools, and I showed some analytical tools that we will leverage here in this example that allow us to find differences in composition and allow us to understand the relationship between different species in terms of their abundance or richness. Once you have that, you also rely on databases. So for example, with 16S ribosomal RNA sequencing, we do rely on different databases for annotation. Ultimately, your goal is to understand the relationship between the statistical analysis and the biological interpretation that you can develop for specific features. So we will go through some of those steps during this program as well.
Thank you so much.
Yep, thank you for joining. What is
Please,
the weekly time commitment for this course? Okay, yeah, did you have a question, Bello?
yes, I do. I have a question.
Yeah,
Now,
go
for
ahead.
a beginner who is interested to learn microbiome sequencing, could you please suggest the best platform to learn programming language? I mean, maybe someone wants to learn how programming language. Do you have any platform where someone can learn that?
Yeah, so as I already mentioned, we have a whole set of exercises that are designed around data science topics in both R and Python. So you can explore some of those after you create an account on the educational portal. You can actually explore some of those right here. And again, what we start with is we start with the logic of analysis, but then also in many cases we provide you with pieces of code that you can start learning how to modify to apply to a particular data set of your interest.
Okay, thank you.
Yeah, thank you. Yes, there's another question from Srinivasan. Okay. What is the weekly time commitment? So you can look at the syllabus. It depends on whether you would like to have just the training sessions, which is one option. So for example, it's an hour and a half interactive session that will be recorded and all stored in a single location. So you could review all of the sessions at any time. But you can see that that is 2 weeks, 3 sessions per week. So you can see August 31st, September 2nd, and September 4th, that's week 1, and then September 7th, September 9th, and September 11th, that's week 2. And then we transition into a project. So you have a project, how to find the data, how to structure your project, how to start your, translate your idea into an analysis, step-by-step analysis. And then after you have some time to work on your project, and there will be also a review session, you might work by yourself or together with a group. And then at the end, you get feedback from somebody else. Now, this second portion about an independent project is not compulsory. You can get a certificate of participation just after completing the 2-week training, which in total will be 3 1.5-hour sessions in 1 week and 3 1.5-hour sessions in second week. So in a total of 10 hours, you can complete the whole program. Now, as you can see, for each one of these sessions, there are some additional resources, and the minimum courses are Introduction to Metagenomics, Metagenomics 1, and Metagenomics 2. So there are 3 courses. You can complete each course in about a couple of hours, but they do include some practical assignments that will take a little bit more time. So I think that a good estimate is about 20 hours for the whole program is a good estimate for what you would need. And this is over the course of 2 to 3 weeks, but you will have access to all of these for about a month. So you can actually spread it out throughout the whole month. Can we get access to one of the best proposals? Yes, I believe that Bipsa already mentioned that she will share that with you, but I will also share that now so you could all take a look. So actually, we have a YouTube channel. Thank you, Bipsa. So there's a link. We actually have a YouTube channel where you can find a lot of these presentations for different students. Here's an example of a project related to different topics. So here's a couple of presentations, and on that link that Bipsa provided, there's some more. You will get 2 weeks to complete your project. So from the end of second week all the way to the to October 1st or October 2nd, you will have time to work on your independent project. October 2nd. Okay, great. So for any additional questions, please feel free to reach out to us. marketing@pine.bio is the email address. So Bipsa, you could please put that on the chat as well. This recording is going to be available on YouTube after this. About a project, is it possible to do a group project of a friend or a colleague of mine is interested? Yes, you have to be participants of the program itself to get the feedback and get evaluated if you would like to do that. But within the program, you will have a number of different opportunities to interact with others. And we have many examples where people have worked together to produce group projects. So I'll just give you an example of a group project that you can see. So you can see an example of 3 participants working together and developing a project together. Again, thank you everyone for joining today. If you would like to stay in touch, you can email us to marketing@pine.bio. And for those of you interested in joining the program, we're going to be starting on August 31st. So we're excited and ready to see you there.